In this video, we're going to go over the reabsorption in the proximal convoluted tubule. Proximal convoluted tubule, remember, that receives a filtrate from the renal corpuscle, and it's the first part of the tubule system. It receives a filtrate from the renal corpuscle, and you want to note this. It's the greatest area for reabsorption of substances into those paratubular capillaries. Here, the proximal convoluted tubule reabsorbs virtually all the glucose, amino acids, vitamins, other organic nutrients, a whole host of various ions, and about 65% of the sodium chloride and water, by osmosis, is going to be reabsorbed here in the proximal convoluted tubule. So we're going to see the glomerulus as a filter. Substances come through the glomerulus. The smaller substances, such as the ions, amino acids, glucose, move through the glomerulus into the filtrate. And then in this first part, the proximal convoluted tubule, virtually all the amino acids, all the glucose, a good portion of the sodium, and many other ions are going to get reabsorbed here in the proximal convoluted tubule. Most of the movement of solutes and water is going to be facilitated by active transport of sodium out of the tubule cells at the basolateral membrane shown here by the sodium-potassium pumps. And it's going to pump that sodium out. Remember, 3 sodiums out for 2 potassiums in, and that sodium will then go out to the interstitial fluid here and then into the paratubular capillaries. It's extremely important, as I said before in the previous video, the movement of a lot of these substances through the apical membrane from the lumen of the proximal convoluted tubule moves into those cells by secondary active transport. And sodium is the driving force in that secondary active transport. So you have to keep sodium low here in those cells. And how do you do that? In the basolateral surface, you have these sodium-potassium pumps that are pumping sodium out to keep sodium levels low here so that it can co-transport with glucose, vitamins, other ions, amino acids, can move through by secondary active transport. So the lowered sodium concentrations within the proximal convoluted tubule cells and higher concentration of sodium in that interstitial fluid provides the means for movement of substances through the apical cell membrane, as I just described in the previous slide. You have symport transport processes, you have antiport transport processes, and then you also have transcellular transport here in that proximal convoluted tubule. With the symport, we have chloride ions, calcium ions, amino acids, glucose, phosphate are transported with sodium from the lumen into the cell. Shown here in the proximal convoluted tubule cells, here's sodium moving in, and it's going to move with other substances. The symport process of moving with other substances. But in order to have sodium move down its concentration gradient from the lumen into the cell to power this movement, this symport process, you have to keep sodium low inside that cell. And the way you do that are by the sodium-potassium pumps seen here at the basolateral membrane. Once those substances come through with sodium, then they're going to diffuse out of this cell into the interstitial fluid and then finally into those paratubular capillaries by diffusion. Facilitated diffusion, really, because you're going to have to go through another transport protein. Also very important in the proximal convoluted tubule is that you're going to have the movement of hydrogen ions out into the urine. And it does this by an antiport transport process with sodium. Remember, antiport is in opposite directions. So as sodium moves down its concentration gradient into that cell, it's going to power the movement of hydrogen out of the cell. And this hydrogen that moves out has a major role in the reabsorption of bicarbonate. Because we're going to see that that hydrogen is going to combine with bicarbonate to make carbonic acid, which is going to dissociate into carbon dioxide and water, and you're going to reabsorb the carbon dioxide and water into the cell in order to bring in the bicarbonate. And a really neat process. And we're going to go over this process a little bit later on when we talk about acid-base regulation within the kidneys. You also have transcellular transport, or at least 3 ions, potassium and calcium and magnesium. They can literally diffuse between the adjacent cell membranes. So remember, we talked about the tight junctions shown here. These tight junctions are permeable to certain ions in different places along the tubule system. So they're not as impermeable as in other places of the tubule system. Here in the proximal convoluted tubule, certain ions can move between the cells, what we call that transcellular transport. The increased sodium chloride concentration in those paratubular capillaries here, as you transport the salt from the lumen into the interstitial fluid and then into the paratubular capillaries, this creates a very high osmotic gradient for the movement of water out of the lumen into the peritubular capillaries. About 65% of the salt in water is reabsorbed in the proximal convoluted tubule. You want to star this. And remember that water osmotically follows solutes. You have that saying, water follows salt, water follows solutes. As you pump the solutes out of that proximal convoluted tubule, water is going to follow by osmosis. So you have the reabsorption of major solutes by the proximal convoluted tubule. Here in the apical cell membrane, you have the symport transport with sodium. And there are a number of different substances that you see here that are transported with sodium. Previously, I talked about a few of them. But if you look at it in more detail, all along this apical cell membrane, that sodium gradient, sodium moving down its concentration gradient into the proximal convoluted tubule, provides a driving force to move all of these substances into the proximal convoluted tubule cells. And then once you get to the basolateral membrane, many of those substances are going to move through by facilitated diffusion that you see here at the basal membrane. I'm not expecting you to memorize all of these substances that are moving through in the proximal convoluted tubule. What you should get a general idea of is that when we say that most of the substances are reabsorbed in that proximal convoluted tubule, that there is a lot of substances shown here that are transported across. And the primary driving force for the movement of these substances is secondary active transport with sodium. And then water is going to follow by osmosis. Let's focus now on the reabsorption of glucose and amino acids. We're going to focus on glucose, but amino acids are moving through in a similar fashion. So with glucose and amino acids, these are only reabsorbed in the proximal convoluted tubule. It's the only place where we have transporters for glucose and amino acids, which means if the concentration gets so high with glucose and amino acids that it overwhelms those transporters, you're not going to transport the glucose and amino acids back into the blood, and they're going to be lost in the urine. Well, when you look at glucose amino acids, the way that they're transported is by secondary active transport, which is mediated by membrane carriers that co-transport glucose and sodium, or amino acids and sodium, into the proximal convoluted tubule. So remember, in secondary active transport, ATP is not directly used with this co-transporter here. So we see in the lumen, sodium is high in the filtrate and lower inside the cell. Glucose is low inside here in the lumen and higher inside the cell. Glucose is moving against its concentration gradient, whereas sodium is moving down its concentration gradient. Well, that sodium moving down its concentration gradient is providing the energy to move glucose into the cell by co-transport. Well, this is secondary active transport. So remember that in secondary active transport, you use ATP, but it's used indirectly. So where's the ATP being used? Here's that sodium-potassium pump on the basolateral membrane. In order to keep sodium low here in the proximal convoluted tubule so that sodium continues to move down its concentration gradient, you have to pump sodium out here. You're moving 3 sodiums out for every 2 potassiums in. Remember, sodium is higher here in the interstitial fluid outside the cell. So you're moving sodium against its concentration gradient, and that requires ATP. That ATP provides the movement of sodium out to keep sodium low inside the cell, so that this sodium in the lumen of the tubule can continue to move down its concentration gradient and power the movement of glucose from the lumen inside the proximal convoluted tubule. So is ATP used in secondary active transport? Absolutely. It's just used in a different spot. Well, that glucose that comes in is now higher inside this cell, so it's going to diffuse out by facilitated diffusion out of the basolateral membrane and into the peritubular capillaries. The membrane carriers can achieve a maximal transport rate, called transport maximum, or TM, because you only have so many carriers in the apical membranes of these proximal convoluted tubule cells. If you have so much glucose in the filtrate, you can saturate these to where it reaches the maximum transport that it can transport that glucose across. And they're saturated. You can't transport any more glucose. It's coming in as fast as it can. At that point, if that glucose gets through the proximal convoluted tubule and into the descending limb of the loop of Henle, there are no more transporters for glucose, and it's going to wind up in the urine. So you want to make sure you star that transport maximum, or TM. That occurs when all the membrane carriers become saturated with glucose or amino acids due to excessively high concentrations. This results in excess glucose and or amino acids being lost in the urine. When you have glucose in the urine, we call it glucosouria. This results when more glucose passes through the tubules than can be reabsorbed. So in diabetes mellitus, which is caused by either an inadequate secretion of insulin, which is known as type 1, or you have an inadequate action of insulin. The receptors aren't responding to insulin. We call that type 2 diabetes, both of which can result in hyperglycemia and glucosauria. So the person's going to also excrete large volumes of urine because excess glucose that is not absorbed from these renal tubules carries water with it as a result of osmotic pressure it generates within the tubules. Because remember, in order to reabsorb water, you have to increase the solute concentration around the tubules so that water can move out by osmosis. Well, if you have excess glucose in here in the tubules, you're not going to reabsorb as much water because you're going to create a higher osmotic gradient inside the tubules than is normally there. And the person loses a lot of water. So next up, we're going to be going over the transport mechanisms that happen in the loop of Henle, or loop of nephron.